Immunotherapy wins against breast cancer. Women with a hard-to-treat form of breast cancer were found to live longer when treated with a combination of chemotherapy and immunotherapy drugs. Triple-negative breast cancer is an aggressive form of the disease in which cancer cells lack receptors for estrogen, progesterone, and HER2, making it unresponsive to certain treatments. Cancer cells can be treated with chemotherapy, but often develop resistance to it, allowing them to spread to other body parts. Patients usually survive 18 months or less. A new study found that treatment combining chemotherapy with the immunotherapy drug Tecentriq were more effective, improving overall survival rates by almost 4 months. In the clinical trial, 902 women treated at 246 medical centers in 41 countries were randomly assigned either Tecentriq and chemo or placebo and chemo. Chemotherapy was given once a week while the immunotherapy drug was administered intravenously every 2 weeks. The drug deactivates a protein in the cancer cells which keeps it safe from the immune system, while chemotherapy roughens up its surface, enabling the immune system to recognize and attack the cancer. The new treatment is currently being reviewed and may well become the first FDA-approved immunotherapy drug to treat breast cancer. Cure for cancer? We're getting there.
Metastatic cancer could be stopped. A research team led by the Georgia Institute of Technology has developed a new treatment that can potentially stop cancer cells from migrating inside the human body. Cells contain cytoskeletons to give them their shape and to carry out functions such as division and movement. In order to move, the cytoskeletons produce protrusions, called filopodia, which extends from fibers inside the cell known as lamellipodia. The protrusions help cells to shift locations. Lamellipodia and filopodia are overproduced in malignant cancer cells, enabling them to spread around the body at faster speeds. Cancer kills patients often through the spread of malignant cells, which is known as the metastases process. By attaching nanorods comprised of a small collection of gold atoms to the integrin of the cells, the cytoskeleton can be stopped from overproducing lamellipodia and filopodia, which slows down the migration of malignant cells. A low-energy laser of near-infrared light is then used on the cells. The light is absorbed by the gold nanorods, which then partially melts cancer cells and damages lamellipodia and filopodia. This can bring the migration of the cancer cells to a stop. If desired, the laser light can also be adjusted to kill the cancer cells. The experimental treatment shows no observable damage to healthy cells, which is an advantage over commonly used chemotherapy. In the experiments, scientists also did not see the treated cancer recur.
New vaccine kills cancer in mice. Stanford University researchers used immune stimulators to target cancerous tumors, and it worked surprisingly well. T cells that defend against cancer often end up overpowered by the disease, but scientists find they can be rejuvenated using CpG oligonucleotides and anti-OX40 antibodies. When a microgram of the immune-stimulating agents were injected directly into mouse tumors, the growths disappeared in about 10 days. CpG activates dendritic cells that help against tumor counterattacks, while anti-OX40 antibodies stimulate T cells into attacking the cancer. Once a tumor is destroyed, the cells move through the body to find and eliminate other similar growths. The experiment eradicated lymphoma in 87 out of 90 mice and also worked on breast, colon, and melanoma cancers. The team is looking to start clinical trials to see if the treatment will be as successful in humans.
The pachyderm secret weapon against cancer is a zombie gene. Elephants hardly ever get cancer, and scientists have finally figured out why. Over 17% of people worldwide die from cancer, compared to less than 5% for elephants, even though they have 100 times as many potentially cancerous cells. One reason for this is they have over 20 copies of the tumor suppressor gene p53 that stops DNA damage from turning into cancer, while humans and other animals only have one. Because the cells died as soon as damage was detected, there is zero risk of them ever becoming cancerous. A University of Chicago study found that p53 can also trigger an inactive pseudogene in elephants called LIF6 to come back to life. When cell damage is detected by p53, it activates the LIF6 gene, which in turn produces a protein that pokes holes in the target mitochondria and causes it to die. This genetic resistance to cancer isn't just seen in elephants. Smaller critters like naked mole rats and gray squirrels also have mechanisms that keep them tumor-free. By studying how these animals' genes work, scientists are hoping to figure out how to prevent or treat cancer in humans.
Robot algae wipes out cancer cells. Scientists at the Chinese University of Hong Kong have taken an algae more commonly used as a diet supplement and turned it into a cancer assassin. Researchers coated spirulina microalgae in iron nanoparticles before inserting it inside a rat's stomach. The iron coating, combined with spirulina's natural shape, allowed scientists to control it with magnets. Researchers also put the microalgae inside a lab dish where it eliminated 90% of cancerous cells after 2 days. Spirulina is reported to be toxic only to cancer cells. The biodegradability of the robot depends on how thick the iron nanoparticle coating is. The thicker it is, the longer the robot can operate inside a subject. Lead researcher Dr. Soli Zhang told the publication Science it's not ready for doctors to use, but reckons it might be in about 10 years.
Beating brain cancer with polio. A new experimental treatment is using a modified version of a once-dreaded virus to fight an even deadlier disease. Duke University researchers removed genetic code from the polio virus and replaced it with DNA from the rhinovirus, which causes the common cold. The modified virus is injected directly into the tumors of glioblastoma patients through a catheter. Instead of replicating itself, it attaches to receptors on the surface of the cancer cells, infecting them and prompting the body's immune system to attack. Of the 61 glioblastoma patients treated using the re-engineered virus, 21% are still alive 3 years later, compared to only 4% for those who underwent conventional treatment. The experimental treatment, like many immunotherapies, had varied, sometimes dramatic effects on the subjects. Scientists are still figuring out how to get the virus to produce the same exact results in different people, but are confident that it can be done.
Woman's advanced breast cancer cured with immunotherapy. Could this be a game changer? A woman's advanced stage breast cancer that has spread to other parts of her body was cured using her own immune cells. It marks the first time a T-cell immunotherapy application for late-stage breast cancer was successful. Doctors from the National Cancer Institute in Maryland first took biopsies from the woman's tumors and looked at the DNA mutation specific to her cancer. Physicians then extracted immune cells called tumor-infiltrating lymphocytes, TILs, from the biopsies. Billions of these immune cells were then grown in the lab. The researchers then injected 80 billion of the TILs into the woman's body. The treatment was given in tandem with pembrolizumab, a standard cancer drug that helps the immune system. After 42 weeks, the test showed the woman's body was completely free of cancer. According to The Guardian, researchers are now planning full-scale clinical trials.